Yeah, I'll start now. Good evening, everyone. I'm Dr. Dhanusha. Hope you all are doing good. And I'm here to welcome you all to this webinar on behalf of Hassan Veterinary College as well as Student Alumni Association. And the topic of today's webinar is use of nano trace minerals in livestock feeding. Yes. Next slide. Yes. Now, I would like to welcome Dr. M.C. Shukumar Sir, Dean, Veterinary College Hassan, who's been a very enthusiastic person and a huge support to this initiative. We welcome you, sir. Now, I would like to welcome Dr. Chethan Sir, Assistant Professor, Department of Animal Nutrition, Veterinary College Hassan, who is the speaker of today's webinar. I welcome you, sir. So, with this, I'll start the introduction of Dr. Chethan sir. So, Dr. Chethan sir has completed his graduation from Veterinary College, Bengaluru in the year 2005, and sir has pursued his post-graduation in the Department of Animal Nutrition from Indian Veterinary Research Institute, Bareilly in the year 2008. And sir is currently pursuing his PhD in animal nutrition as well from Veterinary College, Bengaluru. I wholeheartedly welcome you, sir, to this webinar on behalf of Veterinary College Hassan and the Alumni Association Hassan. So, before starting the session, there are a couple of instructions to be followed. All the audience, mute your mic, and the question answer session will be at the end of the session. Please drop your questions in the comment section.
Any other sounds or beep sound? Is that okay?
Sir, make it slideshow.
Okay.
Sir.
Yeah. Okay, right. Okay, is my screen visible?
Yes, sir.
Yes, sir.
Okay, right. Okay, so thank you very much. At the very outset, I would like to thank our Dean, Honorable Dean, for giving me this opportunity, as well as the students of Alumni Association for giving me these opportunities. Okay, so shall I start now? Hello?
Yes, sir, you can start.
Okay, right. So I welcome you all to the nano world, okay, where the next big thing is too small to see. So today's my topic is on use of nano trace minerals in livestock feeding. This is how my presentation will be. So the introduction about the topic, requirement, maximum tolerable levels and relative bioavailability of the minerals, history, development and definition with regard to the nanotechnology, mode of action of these nanoparticles, preparation of nanoparticles, and application of this nanotechnology in animal nutrition. I will be using some of the research reviews and what are the disadvantages of this nanotechnology and risks and regulations with the nanotechnology, future needs, and concluding the topic. Coming to the introduction, so I think we all know the feeding adequate amounts of balanced diets to the animals is one of the important factor for maintenance of good health and optimum production. So in that sense, the minerals play major roles. They're required for various functions in the life process, like for maintenance, growth, production, and reproduction. And even the nutrition status of the animals reflects the productive and reproductive functions of the animals. So in Indian situations or in the tropical conditions, most of our animals, they suffer from mineral imbalance or mineral deficiencies, either due to poor supplementation or because they are exclusively maintained only on grazing or with poor quality straws. So therefore, mineral supplementation can be a what you call at a low cost input strategy for improvement of the livestock production. Another important factor you need to understand here is insufficient mineral supplementation can reduce the performance of the animal with a serious economic impact, while oversupply does not benefit the livestock. and it is damaging to the environment also, because the excess minerals will be excreted in the feces. So that's the reason. So we have to take utmost care in supplementing the minerals, especially with the trace minerals. Okay. So traditionally, the inorganic salts such as oxides, sulfates, and carbonates are supplemented in the concentrated diet as a mineral mixture, and they are fed to the animals. Availability from such sources, that is inorganic salts, is very poor. That means if you think about copper oxide, it is hardly 15% available. If you supply 100 milligrams, only 15 milligram is available. So the cost will also increase. So the scientists, the farmers, even the feed manufacturers, they were looking for some alternative sources of minerals which has better availability. So among them are, one is the chelation mechanism, another is use of organic sources like copper methionine, selenium methionine. So the mineral attached with the organic sources, they have better bioavailability. The problem is the organic source of minerals, though they are very much promising, they are costly. So still the scientists were looking for something else, so which should reduce the cost and which has a better bioavailability and more helpful, so that we can reduce the concentration of mineral supplementation in the feed.
Excuse sir, Chethan
Yes,
sir.
yes.
Please go back, escape, and check the your presentation is not completely visible on the screen.
Okay,
Press escape and check for represent sir.
okay. Is this visible now,
Okay.
or shall I?
Now it's okay.
Okay, shall I present
Slideshow.
slideshow?
Yeah.
Okay, okay. Now,
Sir, it's half of the slide went, sir.
okay,
That has oxides. After that, it's not visible. Carbonates, it's coming. Next, Swami's is also cutted, sir.
okay. Shall I reduce the size of it?
Yes, sir.
Okay, now,
Same, sir.
same. So,
Yeah.
what? Now is it visible?
Slideshow is more than 100%. Check, sir, how much it is.
Actually, it
No,
is.
it's good.
Is it okay now?
Slideshow, sir. Go for slideshow.
Um, Now visible it is.
Next slide. Next slide, sir. Next slides.
Okay.
Yes, now it is good, sir.
Now it's okay.
Yes, sir. Please go ahead, sir.
Okay, right. So I was telling about the availability from organic sources of minerals. Though they are promising, they are costly. So the scientists were looking for some other technology by which we can improve the availability of the mineral with a reduced cost. But definitely they have not come up with a reducing cost, but they have got an alternative where they can increase the surface area, thereby they can increase the availability of the minerals. So bioavailability can be enhanced by increasing the surface area, and this by nanotechnology, where they reduce the size of the minerals into a nano size, will have a higher surface area which will enhance the bioavailability, especially for feeding livestock. So let us see this is, so when you have a higher availability, so I think you can reduce the supplementation in the feed. Okay. So here I have given an example of a cattle, that is gestating heifer, gestating cow, as well as bull. If you see the requirement of the trace mineral, this is as per NRC 2000. This is as per NRC 2001, I think, and requirement is less than 50 ppm. If you take at least the maximum concentration of iron required is less than 50 ppm. When the requirement is so small and the cost is also high, so you can further reduce them, or else you can improve the bioavailability, so which is possible with the nano minerals. And in the introduction I was telling, it's not that you supply more quantity, because these minerals at a higher concentration, they are toxic to the animals also. If you see the maximum tolerable level of the minerals, for copper it is 40 ppm. So if you supply more than this, there will be toxic symptoms. Similarly, for zinc, it is 500 ppm. Manganese, it has got little higher values, that is around 2000 ppm. And selenium, the requirement is only 0.3 ppm, and toxicity is achieved at 5 ppm. So there is a very narrow range. Slight increase in the selenium may lead to toxicity. So that's why, so what I mean to say here is we should not go for supplementing more and more. Instead, improve the availability from the same source so that you can reduce the concentration, and there will be reduction of excretion of these excess minerals in the feces. But in such way, you can reduce the environmental pollution also. So to do all these things, we need to understand what is the bioavailability and relative bioavailability. So bioavailability is, so whatever the thing or whatever the concentration of minerals supplied in the feed which is not appearing in the feces, that means which is absorbed and available for the animals for utilization for productive purpose. Suppose if you say the zinc oxide has been taken as a standard, if you supply 100 milligrams of zinc oxide, 100% all the mineral is absorbed, that is 100%. So if you consider that as a standard, zinc sulfate is 100% available, zinc chloride is 42% available, which means out of 100 milligram, only 42 milligram is absorbed and available for the animals. And if you see the zinc methionine, which is an organic source, or chelated with the methionine, has higher bioavailability, 103 to 133. So by increasing the bioavailability, we can improve the available zinc in the animals for productive purpose. So this is what is relative bioavailability and bioavailability. Similarly, for other minerals also like manganese, so the organic source has higher bioavailability. Similarly, copper, selenium, and iodine. So where the organic sources they have higher bioavailability, so they may require lesser quantity in the feed, and the lesser quantity of copper is excreted in the feces, and lesser is the pollution as well. So this is with regard to the relative bioavailability. So earlier what people were doing is to have a better production, especially with regard to pigs and poultry, they were supplying 20 to 30 fold higher than the normal requirements. Because if you see the energy and protein, they are required in larger quantity. And if you want to increase the energy quantity or protein quantity, then they have to pay more price. But minerals, that too, the trace minerals, though they are required in very small quantity, so if you increase 20 times or 30 times, the price increase in the feed will be lesser. So they were getting better benefits by increasing 20 to 30 folds. But the problem is, so one such example is copper pollution, which is a major problem in the case of pigs and poultry. Especially when these pigs and poultry manures are utilized as the fertilizers, a lot of copper is present in those fertilizers, which will leach out to the nearby streams, ponds, and even the soils. So it is very common in pigs where the dietary concentration of 150 to 250 milligrams of copper as copper sulfate per kg diet, and the zinc sulfate to the tune of 2500 to 3000 milligram per kg diet were supplemented to have a growth promoting effect. Copper also has an antibacterial property. So that's the reason the farmers they were supplying more and more quantity of inorganic minerals. So all these things led to the environmental pollution, and the public become aware about this. And now the public they are bringing regulations, especially when you see the regulations of the European world. There, the fertilizer or the menu which you use for your own farm should not have more than 150 ppm of copper. So such a level the regulations are there, but in India we don't have regulations for menus and all. But still there is a public concern about environmental damage. So I think, and after the end of this session, I think you all come to the conclusion that definitely the nanotechnology approach will have a greater potential, because as it will improve the bioavailability, we can reduce the quantity that we can supply, and thereby we can reduce the quantity excreted in the feces. So let us understand about the nanotechnology, and in that way, so we'll also understand a brief history about this. So Richard Feynman is the scientist who proposed for the first time in the year 1959 the concept of nanotechnology. And in the year 1965, he got the Nobel Prize for proposing this concept of nanotechnology. In his speech, after getting the Nobel Prize, he said, I would like to describe a field in which little has been done, but in which an enormous amount can be done in principle. What I want to talk about is the manipulating and controlling thing on a small scale. At the end, he concluded the Nobel Prize winning speech by saying there is plenty of room at the bottom. So later in the year 1974, Professor Norio Taniguchi, he introduced the term nanotechnology. And again in the year 1999, or Fritas, he wrote a book on nanomedicine. That was the first book which on nanomedicine. And later in the year 2000, National Nanotechnology Initiative was launched by USA. So the nano, the word is actually derived from Greek nanos, meaning dwarf. So how small it is then? So the word nanomaterial is generally used for materials with a size ranging between 1 to 100 nanometers. So if I say 1 to 100 nanometers, you may not be able to imagine this. So to understand better picture about the nanoparticle, so you can see here different pictures are there. There is a human being standing there, a blood drop is there, a blood cell, a DNA strand, many other things are there. Okay, so I think in this picture you will have a better idea about the nanomaterial. A human being or a person with 6 feet tall will measure around 2 billion nanometer. An apple will measure around 80 million nanometers. When you say micro, our human hair is 75,000 nanometers, and your E. coli bacteria is 2,000 nanometers. These are not visible to eyes, especially the bacteria. So the smallest the eye can see is 10,000 nanometers. Imagine what is the size of nanoparticles. So our DNA strand is 2 nanometers. The buckyball, which is made up of single layer of carbon, is 1 nanometers. And your touch screen, which is made up of graphene, so a carbon nanotubes or a graphene buckyball, so they measure 1 to 1.3 nanometers. I think now you can relate. So what will be the size of nanoparticles? So let's understand what is nanotechnology. The nanotechnology initiative by US molecular and macromolecular levels using a length scale of approximately 1 to 100 nanometer in particular particle to the tune of 1 to 100 nanometer. At this scale, the physical, the chemical, and sometimes unexpectedly from those of the corresponding bulky materials. Currently, this nanotechnology
ಯಾಕೋ ಶಿಟ್ ಎಷ್ಟೊತ್ತಾಯ್ತು
ಈಗ ಕರೆ ಬಂದಲ್ಲ
training by fenestration. This will also enable efficient uptake by the cells. And these nanoparticles can also be used for efficient delivery of the active compounds to the target sites in the body because they are able to pass or penetrate deep into the tissues. So because of all these properties, these nanominerals they can show or they show their significant effects even at lower dose of recommendation than the conventional mineral sources. So suppose a copper is required at 40 ppm, as a nanomineral it may require only 10 ppm or 20 ppm to fulfill the same function. These nanominerals they have significant growth promoting, immunomodulatory, antibacterial effects than the conventional counterparts. Same mineral, just the size is reduced. They have a different effect. Apart from this, the nanominerals are reported to enhance the reproduction in the livestock as well as poultry. What other properties of nanoparticles which give a different effect than the conventional or the bulky compound of their same origin? They have a changed stability, they have different quantum effects, which I will explain later. They have surface area effects, and even the shape of the nanoparticles will have a different property than the original one. The shape can be a spear shape, or a spherical, hexagonal, square, cone shape, or dendritic shapes. They all have, based on their shape also, the property will vary. So when you reduce the material from macro into nanoscale, they behave differently. They have a quantum effect. Some of the example for this quantum effect is the copper, which is opaque at the room temperature in a normal sense, in a macro scale, will become transparent when you reduce it to nanoscale. Platinum, which is a inert material, will become catalyst. Aluminium, which is stable, will be turn combustible. Gold, which is solid at room temperature, will turn into liquid. Imagine your gold in liquid state. Silicon, which is an insulator, becomes conductor. I think you might have observed, studied the silicon conductors, chips, which now we used. So all in nanotechnology, where they have reduced the size. This picture will give a better uh, knowledge with regard to change in the property of the material when you reduce the size to nano. So gold, if you see the yellow metal, when you reduce it to nano, it becomes transparent, it becomes liquid, and it turns red in color. It is not yellow when it is reduced to the size of nano. And this property is not new to us. In the year around 1820 to 30, the churches in the Europe they have used this nano gold for painting the glasses with red color. They have used even nowadays the nano technology or property is used in cosmetics, medicines, human medicines, many other electronics, computers, even your paintings, zinc oxide, nano zinc oxide used in your paintings. But I've observed one of the advertisement where Ranbir Kapoor and Priyanka Chopra, they say the wall is antibacterial. That is due to the nanotechnology of this nano zinc oxide. And now let's understand about the bioavailability. As I said earlier, so whatever you feed and which is not appeared in the feces, it should be assumed or presumed that it is absorbed. So if your mineral is absorbed 100%, so it has a better bioavailability. We don't use digestibility in the case of minerals. The reason being, most of the minerals which are utilized in the body and they are excreted back into the gastrointestinal tract through bile. So that's why, so what we use the term in the case of mineral is bioavailability. That is which is absorbed and not appearing in the feces. So when you reduce the size at the nanoscale, they have bioavailability higher than any other conventional mineral source. And this nanominerals, they improve the bioavailability. The reason being is due to increase in the surface area. Though there are many other factors which affect the bioavailability, like the animal species, the physiological status, previous nutrition, solubility of that particular mineral element, or interaction with any other dietary nutrients or between the minerals. But due to increase in the surface area, there will be increase in the bioavailability, which happens in the case of when you reduce the size of the material into nano scale. So let's briefly understand about this preparation of nanoparticles or synthesis of nanoparticles. There are mainly three methods by which these nanoparticles or nanominerals can be synthesized. One is by physical, another chemical, and biological method. Within this physical, we have mechanical method, physical vapor deposition, gas phase synthesis method, and in chemical we have microemulsion method, precipitation method, and green chemical synthesis method. In the biological method, use of microorganism as well as plant material or extracts. Among the three, safest and environmental friendly is biological method, and the cheapest is chemical method, but it will leave the hazardous chemicals. So thus most of the people they do not prefer for going chemical method. Even the person who involved in the synthesis of these nanoparticles may get some hazards, chemical hazards. And most commonly used is physical method, that is mainly the ball milling method. This physical method is the mechanical grinding of bulk metals with subsequent stabilization using volatile protecting agent. Among the physical method is the ball milling. Ball milling method is the most commonly used. And in the chemical methods, here we use chemicals for reduction of silver salts using reducing agents like borohydrate and 2-mercaptoethanol. In the biological methods, we use microorganisms like fungi, yeast, bacteria. Some of the examples of using microorganism is pseudomonas used in preparation of silver nanoparticles. Lactobacillus is used in synthesis of gold and silver nanoparticles. Similarly, one of the plant commonly used for this extract in Kannada we call it as paneer patre. The crushed leaves of geranium plant is used to synthesize gold nanoparticles. These are some of the methods by which they synthesize. I will not go in deep about the process and how they prepare this. So let's understand what is the effect of these nano minerals in animals. Some of the research has been already going on. So I have taken some of the reviews and research papers and their findings to better understand the effect of feeding nano minerals. Very first, we'll understand about nano copper. UGR, he is the scientist who has studied this, and he has studied effect of nano copper on copper availability, growth performance, and serum traits of piglets. He used 36 piglets of 4 months age in 3 treatments. Okay, so in the first treatment, it is the basal diet control. Another, he has used inorganic mineral copper sulfate at 50 milligram per kg. And the third group is nano copper, the same 50 milligram per kg feed. And he did his experiment for 47 days. After 47 days, he observed, especially with regard to growth performance, there was no much difference in the initial body weight and final body weight, but the average daily gain was significantly better in the case of nano copper supplemented groups. You can see, so the average daily gain is 656 gram. Similarly, the FCR is also improved, that is 1.50, and copper availability is 44%. So significantly higher than the inorganic as well as control group. And another important enzyme which is having copper as its cofactor is superoxide dismutase, which has antioxidant role, and this concentration of this enzyme is much higher in nano supplemented group. And he also observed what is the concentration of copper excreted in the feces. In the nano copper supplemented group, he could observe lesser copper content, that is only 88.75 milligram per day. Whereas in the inorganic copper, at the same level, 50 milligram per kg, he could find that 104.6 milligrams of copper was excreted. So he could reduce some copper in the feces, so thus even improving the bioavailability of this copper. Another study by Wang and his co-workers, where they have studied the effects of copper nanoparticles on growth and immunity in broiler chicken. They have used around 200 biologics divided into four groups, and the study was conducted for 42 days. The first group, the control group, is with basal diet with zero supplementation of copper nanoparticles, and he has used 50, 100, and 150 milligram per kg of nano copper. I think his hypothesis is to find out at what level they are more he can achieve best growth. At 100 milligrams, he could achieve better results. That is ADG of 49.3H and FCR of 1.96 at 100 milligrams of nanocopper. And even the immunological parameters, the serum immunoglobulin were much higher in 100 milligrams compared to the control groups. So all other 50, 100, and 50 were comparatively higher than the control, but among these three, the 100 milligram per kg nanocopper supplement gave better results. And he also found out the population of lactobacillus and bifidobacterium in the cecal digesta were increased, and the population of coliform, which are bad bacteria, was decreased. And the dietary supplementation of nanoparticle copper, increased the serum total protein and albumin, whereas decreased urea nitrogen, which is a beneficial effect. So coming to the effect of feeding on nano zinc in animals. So this study was conducted by Mishra and co-workers in the year 2017. The effect of supplementation of nano zinc on zinc bioavailability, immunomodulation in female Murrah buffalo cows. He used 20 female Murrah buffalo cows divided into four groups, and his study period he conducted study for 120 days. So the basal diet, which contained 21 ppm of zinc, there was no additional supplementation, and zinc sulfate group, that is 40 ppm, and he used nano zinc at two levels. One is at 10 ppm and one is at 20 ppm. And he could find out that at 10 ppm and 20 ppm supplementation, they were on par with the 40 ppm inorganic zinc supplemented groups. So you see the zinc retention, which is 55.20 in 20 ppm nano zinc and 55.89 in zinc sulfate group. So that means if you supply only 20 ppm, it is on par with the 40 ppm. So suppose if you, instead of inorganic source, if you use nano source, definitely you can reduce the concentration of zinc supplementation, thereby reduce the excretion. Similarly, other parameter like serum zinc level and most of the parameters are on par or even better than the inorganic supplemented groups. So the nano zinc at 20 ppm is performing similar to the 40 ppm inorganic source. So another study by Rajendra and co-workers in the year 2013, where they have studied the effect of nano zinc oxide on milk production and immunity in Holstein Friesian crossbred crows. So they could find out there will be increased milk production at nano zinc at 60 ppm. They have used three different sources. One is zinc oxide, inorganic source, zinc methionine, the organic source, and another is nano zinc at 60 ppm. And they could find out that there will be increase in milk production. Highest amount of increase was found in the zinc, nano zinc supplemented group. Similarly, the somatic cell count was much lesser in the nano zinc supplemented group because the zinc has the antibacterial property and thus reducing the inflammation. So the reduce in the somatic cell count. Similarly, serum zinc level was also higher in the nano zinc supplemented group. And some other researchers they also found, one such example is Zhao and co-workers. They have reported that supplementation of 20 ppm nano zinc in broilers significantly improved weight gain, feed efficiency, total antioxidant capability, and as well as SOD and catalase activity compared to the control fed 60 ppm of zinc oxide. Here they have compared inorganic versus the nano zinc. Inorganic was supplemented at 60 ppm, whereas nano zinc was supplemented 20 ppm. So we can reduce the supplementation dose. Another work, Siddhartha, this was conducted in Bangalore, about Veterinary College, Poultry Department. He studied different sources of zinc on growth performance, carcass traits, and immune status in Giriraja birds. He used zinc sulfate, inorganic source, zinc methionine, organic source, and nano zincs. He could find out at 60 milligrams per kg of nano zinc found improved body weight gain and feed conversion efficiency, and even the serum and hematological parameters were better compared to the inorganic and organic sources. Even the carcass dressing percentage was also better in nano zinc supplemented group. So Chen and co-workers, this is a in vitro study. So they have studied on the effect of this nano zinc supplementation on rumen fermentation parameters, and he has supplemented 0, 50, 100, 200, 400 milligram per kg diameter, and overall he could find out that the growth of ruminal microorganism, there was increase in the ruminal microbial protein synthesis, the raise, and also there is raise the energy utilization efficiency in early phase, that is 6 to 12 hours of incubation in vitro. And he could also found out that there was increase in the concentration of volatile fatty acid, microbial crude protein production, and fermentation of organic matter in the nano zinc supplemented group. And overall, he could conclude that at 100 and 200 milligram per kg of nano zinc oxide, there was better results, like decreasing the concentration of ammonia nitrogen, and there was increasing the propionate ratio compared to other groups. So coming to effect of feeding nano selenium. So selenium, so effect of selenium, nano selenium and selenium yeast in feed digestibility and rumen fermentation in sheep. So here they have used nano selenium and another is organic source, that is selenium yeast. And this study was conducted on 18 male sheep, divided into 3 groups. The control group, another is supplement with 3 milligrams, and that is nano selenium, another is 3 milligrams of selenium yeast. And they could found out that the nano selenium group performed better, and especially with regard to the nutrient digestibility, the dry matter, NDF, ADF, protein, good protein, all the parameters, the nano selenium group performed better than the even the selenium yeast as well as the control group. Even the rumen parameters were also comparatively better in nano selenium supplemented groups. So another study, effect of nano selenium on semen quality. As you know, the selenium is an important immunomodulating and having a better effect on the reproductive ability, glutathione peroxide activity, even the testis ultrastructure in male boer goats, which they have studied, and they have used 40 male black goats and the age of around 3 months, and 4 treatments where they used sodium selenate, which is an inorganic source, yeast selenium, organic source, and nano selenium at 0.3 ppm. And all the parameters like average daily gain, the final body weight, blood selenium concentration, serum selenium concentration, liver concentration, even the antioxidant properties like SOD, so glutathione peroxidase, as well as sodium um, SOD activity, enzyme activity were much higher in nano selenium supplemented groups. Another study by Shia and co-workers in the 2011, effect of nano selenium on semen quality. Of course, it's the same. Semen characteristics. This was on semen characteristics, and they could find all the parameters like semen antioxidant property, ATPase concentration, testicular glutathione peroxide concentration were higher, and sperm abnormality rate was much higher in control group than the nano selenium groups. This they have used only 2 treatments. One is control, another is nano selenium. So the same study in the case of broilers, the comparative effects of nano selenium and sodium selenate on growth in broiler chickens. They have used inorganic at 2 levels, 0.15 and 0.3. Similarly, nano selenium at 2 levels, 0.15 and 0.3 ppm. And they could find at 0.3 ppm. So there was better performance, especially with regard to average daily gain, serum glutathione peroxide concentration, serum selenium concentration, even in the liver, kidney, and muscle. It has a better concentration. And with regard to the feed efficiency, the inorganic as well as nano was similar. Feed efficiency was similar. but better than the control group. But all other carcass characteristics as well as the concentration in the organs was much higher, which is a beneficial effect. They have also the zoo and another study. They have observed that glutathione peroxidase activities in carp, that is a fish plasma, and liver of nano selenium group and selenomethionine. Selenomethionine is an organic source that significantly improved with that of the control. So organic and nano they are performing better than the control. Another study, nano selenium should be most effective as a chemopreventive agent at smaller particle size as it improves the activities of glutathione S transferase. So even immune boosting activity is better when you supply the selenium as nano selenium. Now, effect of feeding nano chromium. Chromium is mainly used, especially for improving the carcass characteristics, especially in the case of pigs, the carcass characteristics and pore quality. They have supplemented at 200 microgram of nanochromium in finishing pigs, and they have found out that higher carcass lean percentage, larger longivorous muscle area, lower carcass fat percentage, and lower back fat thickness. All these characters will fetch the pork at a higher price as per the consumer need. The drip loss in chops from pigs fed to nanochromium was decreased by 21.5%, and weights of the longissimus and semimembranous muscle were increased by 16 and 15% respectively. So, in addition, supplementation of the nanochromium resulted in 184, 145, 88, and 53% increment of chromium concentration in this muscle, liver, kidney, and other organs. So, improved concentration were also found. Another study is supplementation of chromium yeast, which is a organic source, and nanochromium in Giriraja bird. This study was conducted by Malathi in the year 2015. This was her thesis, PhD thesis. The chromium yeast at 200, 400, and 600 ppb levels, and nanochromium was supplemented at 50, 100, 200, and 400 ppb levels. And they found that the cumulative body weight, feed intake, and FCR were not affected, but the increased serum levels of total protein, albumin, globulin, and antibody titer against NDV and IBDV, hemoglobin percent, chromium content in the blood and tissues were improved. And so nano supplementation produced lean meat and reduced serum triglycerides, cholesterol, glucose, and abdominal fat. In the trial 2, the same person has carried out supplementation of chromium yeast and nanochromium in Giriraja bird and found out the increased egg production, egg weight, albumin index, fat, and cholesterol content in the eggs. Thus, meat and egg were enriched with nanochromium, and this can be used as functional food by patients with type 2 diabetes. As you know, the chromium will enhance the performance of the insulin. It is also called as glucose tolerance factor. So, thus, the meat enriched with chromium can be used by type 2 diabetes patients. So, coming to effect of feeding nano iron, you all know iron is very much important, especially in the case of piglets. So, iron is normally supplemented as ferrous sulfate, but it has inconvenience in giving food a metallic taste. So, if you paste or smear the iron or ferrous sulfate on the udder of the mother, piglets may not like to eat it. It gives a metallic taste. Nanoparticles of ferrous phosphate are highly available, making it more useful than its normal form. Okay, so the nanoparticles, they can also bypass the physiological ways of nutrient distribution and transport across the tissue and cell membranes, especially in the sow. Because we all know iron cannot pass through the placenta and available for the piglets. In nano form, it may be also possible. Nano dispersed forms of iron supplementation to piglets showed hematological values were within physiological norms. That means there was no harm, because once iron is absorbed, it will not be excreted out. So if you supply in the nano form, it may get accumulated. This was a hypothesis, but it was not so. It was within the normal values. Efficacy of iron nanoparticles in preventing iron deficiency anemia in pigs was superior, followed by iron dextron injection and oral ferric ammonium citrate compared to these two, the nanoparticle iron was better than the iron dextro injection or oral ferric ammonium citrate giving. So this is all about the effect of these different nanominerals on the performance of the animals. And in the literature, I could not find any of the study which tells me that the nano minerals are harmful to the animals. Either maybe there was no such study, or there may be very less study on the toxic effects of the nano minerals. I could not find any other um, research which gives a negative effect of these nano minerals. But with regard to the nanotechnology, a lot of research has been going on in other areas, maybe in the baby foods, human foods, cosmetics, paint technology, electronics, other things, where they could find some of the disadvantage of this nanotechnology. One, the very first and foremost is enormous financial cost is involved in nanotechnology because it has to come to the market first. When it comes to the market, it will take years to take back that profit or whatever the expenditure incurred. So initially, now if you go for a nano mineral, it will be a much costlier than the organic mineral. Nanomaterials, they can cause damage to membranes, DNA, and organelles through the production of reactive oxygen species as well as reactive nitrogen species. So this is one area where we need to study more. Another important thing is biopersistence. That is, once you feed a nanoparticle, how long it is going to be present in the animal tissue or animal products like milk, meat, and egg. That is another issue. So ionized silver from the silver nanoparticle has been found to be persistent in the organs of animals exposed. So there is what is the withdrawal period? We don't know. Nanoparticles are very light and can easily become airborne and can cause asthma, bronchitis, and can be fatal. Especially people who are working in an industry where they are synthesizing the nanoparticles. It can become an occupational hazard that need to be taken care. Nanoparticles flowing through the bloodstream may affect the clotting system. So with this regard, a lot of research has to done. There are many risks associated with the use of these nanoparticles. As per the research is concerned, we use only small quantity for testing the animals. We don't have so much of risk. But when we use at the industrial level, when we produce in tons of nanominerals, we may have risks which we may not know. The human health and environment, because whatever the fumes, petroleum fumes from the vehicle which is coming out contain carbon nanoparticles. How it is affecting the environment, still we are not able to measure it. And still we know only the short-term results. We don't know in a long term, some 10 years, 20 years, what may be the effect on the animal, what may be the effect on the human beings who consume the animal fed with nanominerals. Still the study has to be done. We don't know till yet. So there are a lot of risks are presently associated. They are not answered now. So coming to the regulations, these are nothing but the rules and legislation that government has to do. So at present, the existing laws are inadequate to assess the risks posed by the nano-based foods. The reason being, toxicity risks remain very poorly understood because of their unique properties, and the number of studies undertaken are also very less. Not assessed as a new chemical according to many regulations. Nanominerals are also treated as any other mineral, like inorganic mineral or organic mineral, but they have a very different properties, which can be unexpectedly a different one. But the rulers or the one who makes policies, they are assessing it as any other chemical, not a new chemical. Even the American FDA is also assessing it as a regular mineral. Current exposure and safety methods are not suitable for handling nanomaterials. We have to develop a higher level of safety methods when you got exposure to the nanominerals. New international regulations of nanotechnology or nanoproducts are yet to come. Even at the international level, many of the countries they have not brought up the regulations. The world over, countries are vying to establish a regulatory framework to address all these concerns. The initiative in this regard comprise enacting new regulations or amending existing regulations or laws for nanotechnology and enforcing safety guidelines for researchers, workers in the laboratories, in the universities, or in the research centers or companies need to be taken up. Government has to publish safety guidelines. Those persons who are handling these nanominerals should follow so and so SOP. And ultimately, we need a global standard which applies to all over the world. Still, it has to come up. So let's come to what is the developments in India. It's happening at world level. Still, the regulations are being framed. But in India, what is going on? A dedicated program was launched in the year 2002, designed as Nano Science and Technology Initiative. This was initiated and implemented by Department of Science and Technology. This will give permission and give funds for conducting research in nanotechnology. In the year 2007, the DST launched the largest and most visible funding initiative to date. That project is called the Nano Mission with a budgetary allocation of 10 billion rupees, which amounts to $170 million US dollars for 5 years. And the Bureau of Indian Standards has been participating in the Technical Committee on Nanotechnology Standards of the International Organization for Standardization. So to adapt for India, it is participating all over the world. In January 2010, the Nano Mission announced the creation of Nanotechnology Regulatory Board, which will supervise what are all the research going on at nanotechnology at different places, and timely it will frame rules and regulations to which we have to adhere. In 2011, of a guidance document for safe handling of nanomaterials in the laboratory has been released by this regulatory board. And this is a picture of India showing where all this nanotechnology research is happening. This is not mainly in the animal nutrition side, but in other areas like electronics, computers, photovoltaics, paints, and other areas. Especially the top institutes involved in nanotechnology research are the IIT Delhi, IIT Kharagpur, Bombay, University of Pune, IIC Bangalore, and many others. So this is with regard to the present situation of regulatory and risk associated with the nanoparticles. So coming to the future needs, what it requires is the optimum levels of nanotrace minerals in the ration that can provide better performance and economic benefits has to be decided. For all minerals, still not yet decided. Thorough and systematic studies are recommended for elucidating toxic effects, dose fixation, and economic production procedures. Regulatory frameworks and various approaches to ensure safety of nanoproducts in agriculture, feed, and food has to be done. India has only taken initial steps in addressing the standardization issues. Okay, so that has to be taken up. To conclude my topic, the nanotrace minerals, they have higher bioavailability than the conventional source of minerals. Studies so far have indicated that the application of nanominerals in animal production, immunity, and reproduction is promising. The safety in application of nanominerals needs to be assessed before it is being applied in the livestock industry. Nanotechnology can be used in animal nutrition sector to improve feed quality, bioavailability of trace minerals, growth, production performance, and immune status in livestock. But the proper legal framework, provisions to be employed for biologically safe, cost-effective production and utilization of nanoparticles for livestock feeding has to be done. However, the work on use of nanoparticles of minerals is still very limited and needs to be taken up. Because if you go to the market for supplementation of nanominerals to the livestock, you won't get any product. You may get for human beings. If you search in the Google, definitely there are some products with labeled as nanominerals. But as of now, for animals for supplementation of nano minerals are not available in the market. So this is a picture I have taken it from Google. So it says Nano Zinc, mineral water with Nano Zinc and copper. It is available, but how safe and what's the cost is very little known. So at the end, I would like to conclude by saying any intelligent food can make things bigger, more complex, and more violent. It takes a genius and a lot of courage to move in the opposite direction. This quote was by Albert Einstein. And thank you for your attention. And if you have any questions, so you can ask. Thank you very much.
Yes, thank you, sir, for sharing the knowledge and the growing trends in this sector. So we have a question from Mr. Shivkumar saying that is there any research on effect of nanocalcium on the milk production in case of cows or the dairy cattle?
So as of my literature survey, I could not find out any. The probable reason maybe calcium is a very cheaper in market and it requires in larger quantity. So I think in future they may take up that also. But as of now, they have tried only in the trace minerals. But there were no research on nano calcium.
Okay, thank you, sir.
Okay.
So there is another question from Dr. Shrikant stating that can we use the nanoparticles parenterally?
Parenterally.
Yes, sir.
Yes, I think some of the medicines, human medicines, they are using it, and for a nutrition point of view, no such studies are there, and we don't know how it behaves when you use it parenterally. But through orally, a lot of studies are there. Parenteral injections, less studies, and definitely may be harmful, or we have not yet fixed the dose also. So it is very risky, but research has to be done on those lines also.
Okay, thank you, sir. So, participants, is there any other questions to be answered? The participants can unmute themselves and ask the questions.
Okay, then sir, we'll go. We'll have the.
Okay, then. Okay. So, I would like to conclude with the vote of thanks. Indeed, it was a great presentation. A big thank you to you, sir, for all the efforts and for the excellent coverage of the topic in today's webinar. Thank you, Chethan, sir.
Thank you very much.
I also wish to express my gratitude towards Dean, Dr. Shivkumar sir, and all the faculty members for providing us the encouragement and a platform for this webinar. Thank you, sir. I also extend my thanks to all the participants for their patientful listening and cooperation. Thank you, one and all.
Thank you very much.
Okay, sir, we'll finish up the meeting. Thank you, sir. Thank you, everyone.
Thank you.
Thank you.